second seminar in the STRiVE seminar series. For those of you who are here for the first time, STRiVE is a special topic network on structural variation in evolution, funded by European Society for Evolutionary Biology since half a year ago about. And the network gathers a global community of researchers that aim to or already are addressing the questions on evolutionary role of structural genomic variation. Within the network, we have been very active so far. We have various activities, but I want to mention two of ongoing activities that you might wish to take part in. One is the special issue in the Journal of Evolutionary Biology on genome architecture and evolution. Submissions are open currently during June, as of now. It might get extended, but please visit the webpage of the network, which is you will find on top here, or contact Marcial Escudero to get more information about it. Another ongoing activity that we are very proud of is the kickoff conference in Vila do Conde, which happens between 8th and 10th of July. Registration is unfortunately closed, but there are still a few poster or attendance slots available, and there is a possibility to register late. In addition, you can ask for travel or attendance support, thanks to the grant that we received from the American Genetics Association. For that, also please visit the webpage or contact organizers Rui and Marta that I put here, their contact information. Before we move on to today's speaker, I just wanted to remind you that the STRiVE seminar series is ongoing every 1 or 2 months with some resting period during the summer and winter vacations. But we have 2 confirmed upcoming seminars in September and October. Ivan Eichler and Nina Terkelsen. We will send information, more information closer to the events through our newsletter. And you can also, of course, visit the website and figure out how to register for our newsletters or to become a member of the network. All seminars will be recorded and posted on YouTube. The first one is already there, so please visit the YouTube page, Structural Variants SDN. Okay, and today I'm very happy to announce that Aurora Ruiz-Herrera Moreno has kindly accepted our invitation to give a talk on structural variation through the lens of the 3D genome. Thanks a lot, Aurora. Aurora is a professor and coordinator of the Genome Integrity and Reproductive Biology SGR group at Universitat Autònoma de Barcelona. She received her PhD from the same university in 2003, after which she traveled for 2 postdoc trainings in South Africa, Stellenbosch University, and another one in Italy, University of Pavia. But thereafter, she returned to her roots and came back to Universitat Autònoma de Barcelona, where she established her research group and was promoted to full professor last year. Congratulations, Aurora. Throughout her research, she was actually involved in remarkable 60 different projects and contributed a large body of knowledge on structural, functional, and evolutionary aspects of genome. And I actually found a remarkable 181 entries on Aurora's Google Scholar profile and impressive 5,100 citations about. Very nice. We are very much looking forward to your talk, Aurora. Please.
Thank you very much, Marina, for the kind invitation. And also thank you for the committee. I mean, I'm really It's a pleasure to be also providing the seminar of what we have been doing in the last year that we find fascinating, and I hope you will also. So let me share the screen first, and then we can try to go. 1 sec. Yeah, trying to put it here.
While you do that, I forgot to say, sorry, to everyone to please post your questions in Q&A. We will read them for you and ask Aurora after she's done with the talk. But if you wish also to speak, you can also raise your hand and we will give you a chance to actually post the question in person. Thank you.
Perfect. You see the full screen, right, Mike? Perfect. So again, thank you very much. It's really a pleasure. So thanks for giving the opportunity to present what we're doing in the lab in this wonderful network that we have assembled all together. And it was nice because I had the options or the chance to give an overview of what we have been doing through these last 15 years or so. It's not going to be too long. Don't worry about that. But it's just to say what our view of looking at structural variations across the tree of life. So most of you, I guess all of you, are interested in biodiversity and which are the different ways that we can understand what we find in nature. And one way, obviously, is to look into the genomes and to look how these genomes are evolving, have evolved, and are evolving through evolution. And when we started to look at these projects that I will show you about, we started from a perspective of chromosomes before all the a huge number of genomes that are available at the moment. We are now living in a really exciting time, but before all genomes were available, we have in our hands chromosomes, right? So we could understand chromosomes under the microscope. And we started analyzing mammalian species, and we realized very quickly, like more than 10 years ago, that there was a huge variation in chromosome numbers across the different phylogroups across mammalian taxa. So we could find species with 100 chromosomes in the Biscacha rats, up to less 6 chromosomes in the Indian muntjac. So it was a huge diversity in chromosome numbers when you compare different mammalian taxa. And but more striking is that when you compare mammals with other vertebrates like reptiles and birds, the situation is completely different. So we have really homogeneous number of chromosomes across taxa, and it's something very different that happened in mammalian. So in the ancestor of mammalian species, what happened was a huge reshuffling of genomes. And this is more evident in this side of the slide, where you can, here we compare full genomes across different representative species of vertebrates. And you could see that most of reptiles, they conserve collinearity across macro and micro chromosomes, but in the ancestor of mammalian species, there was a huge reshuffling. So all breakouts and fuse together, fusions, etc. So that was one of the main questions we wanted to answer. So what was causing this, or what is causing this? And that can be extrapolated to other systems, because we started here with vertebrates, but when you now look on the other pieces of the tree of life, something like that also could have happened. So this is one of the main questions we want to answer. And for that, we have to look also not only at the linear sequence of genomes, but how the genomes are organized into chromosomes. And this is just a way to reflect that mammalian species, we are characterized by what we call macrochromosomes, more or less all the same size, but the vertebrates, they have like 2 types of chromosomes, macro and micro chromosomes. And there's evidence to suspect that this is ancestral configuration for vertebrates. So what happened here in the ancestor of mammalian species is a big question mark over here. So something happened that we have really rearranged chromosomes in ex-ante species when you compare with ancestral genomes, fast diversification in a short period of time that allowed the colonization of new habitats. And that is also reflected in the way the chromosomes look. right? And also the ways the chromosomes are organized within the nucleus of cells. So these images come from microscopy images from works from more than 20 years ago, reflecting that each chromosomes occupy specific positions within the nucleus. This is fluorescence images detecting each human chromosomes in here, and also the same occurs. This is a chicken nuclei. And you could see that what happens is that the macro chromosome, the big ones, are located at the periphery, while the small ones, the small ones are gene-rich, GC high content, are located towards the center. So this is one question that we're really passing. So why this is organization in this way? And probably this organization is affecting the way the chromosomes break, repair, and are organized across different phylogenetic branches. So this is what we try to answer. So when we started this a few years ago, our questions is simple, but really, really big questions to answer. Where organizations occur? They occur randomly in the genome? Is any specific trend that we can adapt? And what drives that? Is that the linear sequence, or also is the way the chromosomes are organized within the nucleus? And this comes from early work that I did during my postdoc. So Marina said that I was in South Africa for the first postdoc, basically because a hotspot of biodiversity, and give us a chance to really have samples from different mammalian species and vertebrates to be able to do all the work that we are doing now, 10 years later. And the first thing that we observed is taking into account also human reference at the human genome as a reference, but at that time there were not many genomes available. So we could observe, comparing the human chromosome 7 in this case with all the genomes from different species that were available at that moment, is that there were huge pieces of genomes that were conserved across taxa, what we call homologous synteny blocks, but then there were regions that this synteny is broken. And you could see that when you compare different species, there are regions that are reuse, that are always break in this, so, and then allow us to reconstruct the ancestor, in this case the Ruiz-Herrera ancestor, to be able to see where in the genomes this reorganization occur. And we realized back then that most of these regions are also colocalizing with high repeating content. This is tandem repeats across the human genome. So we started to see kind of trend across these comparative studies. So what we try to get then is to, okay, can we now compare genomes across different phylogenetic groups, across taxa? Can we reconstruct ancestral genomes? And through speciation time, can we detect lineage-specific reorganizations, for example, translocations, inversions, fusions, and so on? Because that would happen in the ancestor of mammalian species, at least in the case of vertebrates, and not so much in other vertebrate lineages. So in addition to lineage-specific reorganization, and you can do that comparing whole genome sequences, we could also define which regions these reorganizations occur, what we call evolutionary breakpoint regions. So then you can have a map of different taxa, different species, to be able to reconstruct lineage-specific EBRs, or the ones that are conserved. So we have been doing so for many years now. We started with Marta Ferrer. She was one of the trainees in the lab years ago, and we started to work on the ancestral genomes. And since then, we have worked with primates, Afrotheria, rodents, or marsupials. So in a way to have a whole picture of how genomes have been reorganized in the different lineages. And that provides us quite a lot of information regarding what are those sequences that are involved in the reorganizations, or what is in there. So the question, are these regions that are broken are unstable because they have a specific composition of genome organization, or are they just areas where selection is minimal? So as you can imagine, the answer is not simple. And through the years, and not only us, but other labs has been detecting different patterns. For example, the presence of repetitive elements in the regions that are involved in structural variants, like simple repeats, transposable elements, segmental duplications, or so. But not only that, we also detect, and others at the early beginning, that in those regions of breaks, they also have a high percentage of genes. It's a gene density. And in different lineages, there were genes involved in functional constraints. For example, in the case of rodents, we saw genes involved in odorant reception. So adaptation maybe is also playing a role in here. And but not only that, these two factors refers to the linear sequence of genome. Something that we started to think of is like, okay, this is not is only one side of the picture because the DNA is organized in the 3D inside the nucleus of cells. So we add this dimension to the study. So it could be that chromatin structure also is affecting the way chromosomes are reorganized in a lineage-specific way. So this is something that we started to look at more recently. And just to show you very briefly what we have accumulated in the literature about the knowledge about the structure of the genome within the nucleus of cells. So we have nucleosomes organized into DNA loops thanks to cohesins and CTCF. Then we have TADs, or topological associated domains, that forms compartments, A and B compartments that are related to histone modification, so active, inactive, and then we have chromosomes. So we have gone from linear sequences to chromosome position, and then we have something in between, which is quite complex, and that we try to disentangle to see the relationship among this. And we came with the idea some time ago that the way that the chromatin is organized in the three-dimensional space can affect genome organizations. Why? Because when we started to look at evolutionary breakpoints in the early beginning, with the few genomes that were available, we realized that most of the EBRs could be located in TAD boundaries. This is 3 TADs, and this is where normally you have genes and promoters that regulate together. And in most of the studies so far, there was observation that the boundaries are collocated with EBRs. And also that occurs in active compartments, those that they have open chromatin through the cell cycle. So it could make sense that the way you are organized could also determine when you break how you can repair, and you could repair with someone that is nearby you. So that was the idea, and since then what I have been doing is to generate this kind of data through Hi-C. We do Hi-C in situ. We don't use kits because every single species that we have in our hands is kind of different. So we try to troubleshoot the process every time to get this kind of maps, which is contact interaction maps, when you here represent the relative frequency of contact between loci genome-wide. So this is chromosome 1 again itself. So loci that are close by are really interacting, and then these interactions decrease as you go far away in the linear sequence. So with this kind of information, you can extract a lot of data, and we have been doing so with different species. So one of the reasons to go to South Africa is to be able to have access to these nice animals and nice samples. So since then, we have been collecting and constructing or generating fibroblast cell lines from different tissues, from different species, to be able to do these kind of approaches in a large scale. So what I will show you today is just 2 pieces of the projects that we are conducting now, trying to understand patterns of 3D genome diversification across timescales, not only vertebrates, but other taxa. And also because there's a reorganization that needs to occur in the germline to be able to be propagated in subsequent generations across populations. So that's one of the reasons we are also interested in the germline. So these 2 approaches are the main ones that we are conducted in the lab. So for the first one, just a snapshot of what we are doing. This is just an example of the first study that we conducted. Now we are extending this to other species, is to do like Hi-C maps in different species, starting from eutherians, mouse and humans. Then you have Afrotheria, elephant and aardvark, and then marsupials, and then down in the tree to compare with platypus and chicken. Bear in mind that this is Hi-C data generated by the same cell type, somatic cells fibroblasts, which is something important to take into account when you are comparing also these kind of patterns. And I want to draw you the attention not only at the phylogenetic context of the relationship between the species, but also the chromosome number. You could see here that the elephant, mouse, and human, they have between 28 and 23 pairs of chromosomes, but then we have here 3 species with a very low number of chromosomes. So we suspect that, and I will show you why, that's not only that the way that you have the chromosomes organized and the number of chromosomes could also affect all these interactions. That as a control, as I show you in the microscopy images, one thing that you could see, at least in the reptile, is that the microchromosomes interact within each other a lot. And this is something that we could confirm with these heat maps. These maps representing mean interactions between all chromosomes against themselves. So reddish means high interactions and blue is less interactions. So you could see here, like for example, the turtle, alligator, and chicken, the small chromosomes, which are this one. Chromosomes are ordered by size. Here you have interacting more within themselves. This is what we observe under the microscope. So it was nice to see. And this is more or less what happens with other vertebrates like mammals. But you could see here the chromosome numbers. They're really high, from 56 to 78. But what happens with the species with a low number of chromosomes? For example, the marsupials. And what we observe is this. You could see that all chromosomes interact within each other. So this is the same genome size, like 3 gigabase, like this species in here. But then you have small chromosomes that are interacting within themselves. So it gave us an idea that chromatin is more or less intermingled within the nucleus of cells, that might affect somehow the way the chromatin is regulated and is evolving. Another way to see that is this through log interaction maps, where you input here is the inter and intra interactions. So you could see that 3 mammals, they are more or less conserved in terms of the value. But then up here you have marsupials, which means that you have more trans that these interactions, so more interactions between different chromosomes than themselves. And this is the contrary to chicken and other reptiles. They have more condensed chromosomes. So you could see nicely there's different patterns across species. And this is like a summary of the idea. So a species with more or less high number of chromosomes, medium size, they have the chromatin organized into chromosomal territories with DNA loops, and so we also perform ChIP-seq with CTCF to have this. But the nice thing is that in marsupials, which is also a mammalian species, but very at the roots of the mammalian tree, low number of chromosomes, we couldn't see much chromosomal territories as it has been normally described, but centromeres tend to interact within the nucleus of cells, because we also confirm this with immunofluorescence using proteins, centromeric proteins. This is CREST. And you could see that in most of the species that we study, almost of the mammalians, three species so far, we have this structure, wrap-like structure, centromeres towards the center of the nucleus, and then the chromatin at the periphery. And this structure, or like structure, has been described, was described in the literature with insects or mosquitoes, and also plants, that they have a low number of chromosomes. So somehow having your chromosomes in a low number can determine, or will determine, at least the organization that you have the chromatin. And why is this important? Because one nice thing that we saw is that this is the perspective of the chromatin structure, right? But what about the ancestral genomes that I have just discussed at the beginning? One thing that we did is what that was in collaboration with Marta Ferrer, is when we compare, we use all these genomes that were available at the moment, and we reconstruct ancestral karyotypes from marsupials and from Afrotheria. And we detected 2 really nice trends. You could see that the ancestor for marsupials had 7 chromosomes, low number of chromosomes as you would expect, and for Afrotheria, 24 pairs. But what about the structural variants? So it was nice to see that in marsupials from the ancestor, we have more or less conserved the same number of chromosomes, but we detect, in this case, intrachromosomal organization, inversions, pericentric inversions. And nicely, if you just remember, what are the disposition of the chromosomes? Forming this wrap-like structure, like the centromeres are towards the center of the cell. And then one of the hypotheses that we have is that the occurrence of pericentric inversion must be related to the fact that the chromosomes are organized in this way. Having the chromosomes, the centromeres close together. So when you break, you're going to use whatever you have nearby to repair. That's the hypothesis. Because when you compare with elephants and other Afrotheria, this is kind of a mess. We detect inter and intrachromosomal interaction in different numbers, inversions, translocations, fusions, and fissions, and remember that we have chromosomal territories. So in this case, you have different chromosomes with different structures. So that makes that perhaps in this organization, you will have some movement. So it was a nice way to see that perhaps the way you organize the chromatin is affecting somehow the kind of organizations that it could occur. So this is something to have in mind that we are now testing with other species. Another thing that we observe is when we consider inversions, because inversion has been very interesting for many reasons. You all know in this audience that inversion has been involved in chromosome evolution with different models. So we decided, okay, can we look at the structural characteristics and see if there's any difference? So when we compare Afrotheria and marsupials, and when we compare what interactions in inverted regions, we detected like different interactions, and less interaction, more inter-interactions than intermingle. So we came to this hypothesis that perhaps this, what has been described as genomic islands of divergence that we have from the literature, that when an inversion occurs, you have it can facilitate the accumulation of adaptive genes. Here we want to add another level of complexity or another level of information here, the structure. So we have more condensed, more compact chromatin within inversions that could help to add to the process. So this is something that we detected in this mammalian species that we are now trying to convey in others. Another interesting trend, it was like this is reorganization just I showed you before, but when we just observe the homologous internet regions, the regions that are conserved, the question is the conservation at the DNA level also conserved at the structural level? And what you have here is two measures: compartment strength, how strength you have the compartmentalization in your genomes, all the species that you have here, and the boundaries and strength. And you could see that boundaries stats are well conserved across taxa, but compartmentalization change. Compartmentalization is more related to epigenetic marks, active, inactive regions, while TATS is more like the unit of conservation between single promoters. So our idea in this case, and this is something that you could observe here better, sorry. So this is human as a reference compared to mouse, elephant, and Tasmanian devil. This is a marsupial, and these regions are syntenic regions. These are collinear across species, so we now focus on those. And then we compare with the compartmentalization strength, and we detected that obviously the comparisons humans as a reference for obvious reasons, and then you compare with all the species. And what we detected is that as you go far in the divergence time, you lose signal in the compartmentalization. So this is something that you will expect, also determined by sequence level conservation. This is something that we are now exploring, whether this is also related to gene function, regulation of gene function at the compartmentalization level. Because when we compare with TATS in these four species, as you could see, the conservation is higher. So these domains between species are conserved at the DNA sequence levels and also at the TAT level, not that much at the compartmentalization level. So again, we see now different layers of conservation when you compare across taxa, and this is something that it will be nice to see whether this is also the trend for other phylogenetic branches of the tree of life. What else? So this is mammalian species, and you could see here the case of Afrotheria marsupials, but now we also have data from other reptiles like turtles that are also organized into micro and micro, and also some fish, and we could nicely now see whether across the evolutionary tree of life, this kind of organizations are conserved. So compartmentalizations are well conserved at the vertebrate stage, not that much and invertebrates, where we have another kind of organizations across this taxa. And with the idea to try to see whether there's a relationship of phylogenetic signal when you compare this kind of information. For at least in vertebrates, we could see chromosome territories organization, centromere clustering in the case of marsupials, and also centromere-telomere clustering in the case of turtles. So this is revealing that it's a lot of ways or different ways to organize the chromatin within the nucleus of cells. And remember that that could be related also with the diploid numbers. Here you have diploid numbers across taxa, and also to try to see that, for example, in marsupials, we have a low number of chromosomes when you compare with other vertebrates. And so this is what we know so far. Now we're trying to understand other systems, and this is going to be very quickly. We collaborate with Rosa Fernandez here at CSIC in Spain, working with worms. And this is a nice model where you can study the adaptation of animals from water ecosystem to terrestrial ecosystem. And in here you compare the syntenic regions across taxa. You could see a really huge genome reshuffling in the species that are marine when you compare with terrestrial species. So here we have a huge genome reshuffling in terms of adaptation to different ecosystems. But despite this high reorganization, when we compare, for example, OX genes, they tend to be conserved in the same regulatory regions. So despite this high reorganization that might be related to adaptation to different ecosystems, the function of genes that are really well conserved as OX genes are still close together. So we have, again, here different levels of organization. So just to wrap up this first section, in terms of the organization, we have been describing distinct patterns of 3D genome foldings. There's more than one way to organize the chromatin. That could be related to chromosome size, different types of chromosome macro and micro. And in the case of marsupials, we detected this rat-like structure. And in addition to that, we can hypothesize that, at least in the case of inversion so far, they might impose structural constraints in relation to the way the chromatin is organized somehow. I need to disclaimer here is that all the studies I just mentioned are conducted in somatic cells. In fibroblasts, obviously, it's a very specific cell types. It's the ones that we could work with, and other species perhaps is full organisms. But I'm saying that because in order to understand how is this relation coming all together, it's important to understand what happens in the germline, because all these kind of organizations is where the magic happens, no? When chromosomes are organizing in a specific way, we have recombination, we have many molecular mechanisms taking place in here. So this is one of the projects we also are conducting in the lab. So try to understand from the evolutionary perspective how the chromatin or the dynamics of the chromatin in the germline is affecting also chromosomal genome evolution. And just to remind you what meiosis is, so we start from a spermatogonia, a diploid cell, and then we have extensive genome reorganization to have a sperm. Also a disclaimer, for the moment we are working with male germline because it's relatively affordable to work with. We have adults that are producing the germ cell during their lifespan, so it's relatively easy to get. And just to remind you that during meiosis, what happens is a lot of things. So homologous chromosomes organize in a specific way, they find each other, they pair, they break, and they repair through homologous recombination or non-homologous enjoyment. And then at the end, you have recombination, and then you have your germ cells. So when we started this project, nothing much was known of regarding chromatin organization in different taxa. So what we try to understand is whether the way the chromatin is organized is also affecting meiotic recombination, which is really important, and you all know for at least two reasons. This really generates variability, and this is important for the evolution. But something that we also need to take into account is that establish physical connections between two homologous chromosomes during the first meiotic division to avoid aneuploidy. So it's important to get two chromosomes together to really have a successful meiosis, right? So the question always in the field has been where in the genome these crossovers take place. Is there any trend? Is this important for any reason? And it has been one of the big questions in the field because even the early studies, they detected that crossovers are not evenly located across the genome. They are located in hotspots of recombination. And the question is, which factors are affecting this recombination? So what know so far is that it's a pattern. Then the question is, what are the mechanisms that are causing this pattern? And the pattern can be broke into 3 at least. So we have centromeric suppression. Around the centromeres, the recombination normally is reduced. Then you have to ensure 1 crossover per chromosome arm or per chromosome. So the number of chromosomes are important here. But also you have crossover interference. The fact that when you have a crossover, then there's a low probability for a new crossover to occur nearby. So then you have to take all this into account. And the question here is, what determines these genomic distributions across different taxa? Can we set a rule? Can we see different factors? And this is something that we have been interested only related to what I has just mentioned at the beginning of my talk. And for that, we are studying different species, obviously, apart from modern species like mouse and humans. But in the case of marsupials, for the reasons I just told you at the beginning, they have low number of chromosomes, but also they have a funny X and Y. The X and Y in marsupials, they don't have a par. They don't have a pseudoautosomal region because this is something more recent, is innovation in eutherians, not in marsupials. So with this species, we could also even study the origin of sex chromosomes, and also reptiles, because they have different structure of micro and macro chromosomes. They also have sex determination depending on temperature, so they're also interested in that. So just to sum up what we know from all these studies that we have conducted is that really, again, it's not a really simple question when you try to understand what determines recombination across different species, across different landscapes. It could be sequence divergence, it could be epigenetic factors, it could be environmental factors, chromatin organizations, the way the chromatin is organized in the germline, and also chromosomal organization. So this is just to acknowledge the trainees that have been in the lab doing this work. So what I will show you in the next minutes is just 2 pieces of information across these 2 factors, chromatin organization and chromosomal organizations, which are one of the factors that can affect the way you organize recombination. And for that, we need to go back to the germ cells. And in this diagram, I'm showing you the way the chromatin is organized during this process. So what you have with the DNA in early stages of meiosis is that they get anchored to a synaptodermal complex structure, which is a protein structure. It's the way that the chromatin needs to attach to condense. Then this is one homologous chromosomes, and then we have the other homologous chromosomes. So what happens is that when you have these DNA loops, then you have double-strand breaks that they need to repair in the context of the chromosomal axis to eventually have crossover or non-crossover. So why I'm saying that is because this axis, this physical axis of the chromosomes is very important because it determines the substrate of repair or double-strand breaks. And it can determine the final number of crossovers and then recombination. So in the way that length of the synaptodermal complex is a quantitative trait you can measure across cells, across different species, and it's strongly associated to recombination trait, in the sense that the longer you have the axis, more recombination rates you will have because you have more substrate to repair your double-strand breaks. And this correlation is maintained across multiple species or the species that have been analyzed so far. So just 2 pieces of work in this sense. So we initiated this study with the bovids because the question is, okay, if chromosome numbers and the length of the axis determine recombination, can we really prove that? And we studied bovids. Why? Because there's a taxon group that there's a high number of diploid numbers from 30 to 60 chromosomes. So you could see different species across the tree. And from them, we had testes and we analyzed recombination across all of them. We measure the length of the axis and we also measure the length of the DNA loops. And what we found is a correlation between DNA loop size and synaptodermal complex size in chromosomes that were homologous across species. So in the presence of the same DNA content, you will say, as a proxy, the more longer you have the chromosomes, then the loop size is shorted. And then when we analyze the crossover, there's this correlation in the sense that short and long axes, they have low recombination rates when you compare with chromosomes that they have long axes and short, many DNA loop sizes. And basically because there's more substrate in this situation to have a double-strand break resolve as a crossover. So it's really important to take this concept of chromosome structure when we are thinking about recombination in an evolutionary context. It's not an easy task to do because these are really direct estimations of recombination in the germline, but this is a nice evidence that this is correlated. And then you have also correlation between the number of crossovers. MLH1 foci is a specific protein that marks this, with RAD51, which are double-strand breaks. So the more breaks, more crossovers, and then this is related to chromosome size, length, and DNA loop sizes. And related to that, we came with the idea that this structure can have an effect on evolution. So the way that we have different species with different recombination traits, and this is related to structural features, this covariation can also be involved in adaptation. So this is something that we would like also to test in the near future. So can we study DNA loop size? Can we just, instead of going under the microscope and this really tedious work of going cell by cell, can we do that in a more high throughput way? So that's why high-C data is important in this context. And with this, we had a proof of concept where what we did with isolate different cell types in the testes of mice and try to do high-C from all these different cell types to see whether we could infer DNA loop sizes and how the DNA loop sizes are related through spermatogenesis and then how this is related to recombination at the fine scale. For that, we set up like a FACS protocol where we could isolate cells based on DNA content, C to C and 4C, and then nucleus complexity in the way that here you have, in here you have haploid cells, one spermatid and a sperm, and in here you have primary spermatocytes. 4C and then more complexity of the nucleus. So by this way, you could isolate different specific cell types and do all the high throughput analysis downstairs. And this is just to show you how this high-C map look like from fibroblasts to different stages of meiosis. This is prophase 1, and you could see already that the structure changed during the formation of double-strand breaks and crossover. A nice way to see that is by chromosome. This is mouse chromosome 18. This is spermatogonia, prophase 1, 2 phases, round spermatid and sperm. Here we have AB compartment, TATs, whatever is already described, but then in prophase 1, compartments disappear because the chromatin is starting to organize in this way. We had the DNA loops, as I mentioned before, attaching to the complex, synaptodermal complex. So as prophase 1 progresses, the axis gets shorter, and then we have the DNA loop sizes. So fine. So what we have, so this is finishing, or this we have 5 minutes, Marina? Yeah.
Yes,
so,
you have 5 minutes.
Yeah. So just to show you that you could analyze this kind of information up there. And one thing that we observe is that when we do that across the two cell types, and when we compare with the ancestral reconstruction that I mentioned before in the mouse, what we saw is that the evolutionary breakpoints, the regions that have been rearranged during the evolution of rodents, they tend to occur in regions that gets open, that get more accessible in post-meiotic stages, in spermatids. So the idea is that one, that in prophase 1, you really had to do double-strand breaks, really highly programmed, but then in post-meiotic cells, double-strand breaks is not really a constraint. You have really more relaxed selective constraints to accumulate double-strand breaks. So with this, what I want to mention is that it's not only important to study my germ cells, but also that gives us an idea that double-strand breaks, they don't occur randomly. They tend to occur in specific regions of the genome. And this is also important to take into account where, so in which cell type during the process this takes place. So there's a strong selective constraint in here, but not that much in there, right? So let me just, because I'm running out of time. I'm sorry about this, but just let me go to the final take-home matrices. That there's different ways to organize the chromatin. We just started to analyze vertebrates, but probably this could be extrapolated to other taxa. But the chromatin folding in dynamics is chromatin folded during meiosis is highly dynamic, and the way you organize the chromatin really have a way to be related to evolution. And although I didn't have time to show you the chromosome fusions, when they occur, they tend to alter this dynamic. So what we suspect, and it's something that we really like to explore, is that this novelty, new chromosomal interactions that take place in the germline can be a potential for evolution. And just to thank the people that have been involved in the work, present members, and also alumni, and obviously collaborators and funders. And thank you to you for your attention. I'm very sorry that I just ran over time. Thank you very much.
That was perfect. You actually had a few more minutes, Aurora.
So I went very fast at the end. That's fine.
Okay, thanks a lot. Claire, will you stop recording, or will we be...
Yeah, I could stop the recording, or Aurora, do you want to come back in a few minutes about the section you skipped? Or
do I want to go back to
the section you skipped about the structural variants, and maybe we can put a few minutes on that?
Okay.
I mean, we also only...
Okay.
Yeah. We also only have 10 minutes left for questions, so...
Oh, yeah, okay. Yeah. Okay, so I stopped recording and we open the question.
Okay.